Okay, this is week 3 lecture. We're going to talk about the neurophysiology of the neurons, how the neurons are structured, and how they function. Let's go to here. Okay, here we go. Neurons speak to each other. Electrical signals are the vocabulary of the nervous system. Molecules that maintain an electrical charge are called ions. There's two types of ions, cations and anions. Cations have a positive charge and anions have a negative charge. And here we go right now. Let's look at this film. This is video, if I can do this. Okay.
Signals from our environment are most often first detected by the brain. When someone is frightened, a stress response is initiated by their reaction to an environmental signal. But many signals don't stop here. A signal received by the brain can be passed on to many types of cells throughout the body and have widespread effects. Signals get sent around the body by traveling through the nervous system or bloodstream, going from cell to cell. You could think of the cell's sole purpose in life as keeping signals going or initiating new ones in response to the environment. Once a signaling molecule from the bloodstream reaches a cell and docks, the signal is passed into the cell. Like a miniature mousetrap game, the cell is filled with complex contraptions called proteins that bump and nudge each other to keep the signal going until it reaches its target. In this case, it's the production of yet another signaling molecule that will get released into the bloodstream, dock on other cell types, and cause them to react in a variety of ways. The signal-passing machinery inside the cell can look and work very differently, depending on the intended effect. Here we see a signaling cascade that results in the release of energy molecules. Once liberated, they are sent on their way via the bloodstream to power cells in another area of the body. When this tiny domino effect is triggered inside cells, some amazing things can happen. Other signaling cascades triggered during the stress response result in a variety of physical reactions. This signaling molecule can relax or contract different types of muscle cells. When it reaches the skin, the signal docks on muscle cells attached to a body hair. The tiny muscle is stimulated to contract, raising the hair. The signal also docks on cells wrapped around a sweat gland, which contract too, squeezing sweat onto the skin surface. In the lungs, the signaling molecule docks on muscle cells wrapped around bronchioles. But these cells relax instead of contract to allow more oxygen into the blood. Because the protein machinery inside each kind of muscle cell is different, this single type of signaling molecule can cause both relaxation and contraction. Many signaling molecules work this way. When this signal reaches the heart, it docks on beating heart cells. Their response is to beat faster. Blood pressure goes up, and the signaling molecule is rushed on to still more cell types around the body. Constantly and in concert, cells must send and receive a myriad of signals. Cell communication is vital to keeping the body alive.
All right, there you go. Okay. Neurons contain large complex proteins that maintain a negative charge, but because the channels are closed to the larger positively charged sodium molecule, only the smaller positively charged potassium ion can pass through the small channels. Because of the large negatively charged proteins, a neuron at rest maintains a negative resting potential of from negative 50 millivolts to negative 80 millivolts, with an average of negative 70 millivolts. Negative 70 millivolts is the potassium equilibrium potential as equated by the Nernst equation. There are many more potassium cations inside the cell than outside, while there are more sodium cations outside than inside. So the potassium ions or cations are inside, and the sodium cations are outside. When the neuron is stimulated by a neurotransmitter detected by a dendrite, the chemical neurotransmitter causes localized depolarization. In other words, the neurotransmitter causes the sodium channels to open in the immediate area. For a neuron to react, it must receive an adequate amount of neurotransmitter to stimulate enough sodium channels to open up. This is referred to as a neuron's threshold, usually a depolarization of negative 50 millivolts. If the threshold is reached by then enough sodium channels will open to trigger the depolarization. A neuron cannot partially fire, it either fires or it doesn't, and this is known as the all or nothing principle. When the threshold is met, sodium ions flood into that portion of the cell through the rapidly opening sodium channels. This triggers the sodium channels next to the stimulated area to depolarize, as well causing a chain reaction along the neuron until it reaches the neuron's open synaptic cleft. And now we're going to have another, okay, wait a second, okay. Let me see if I can do this. There we go. Okay.
Unstimulated, neurons maintain a constant electrical difference or potential across their cell membranes. This potential, called resting potential, is always negative inside the cell and ranges from negative 40 to negative 90 millivolts. If a neuron is stimulated, the negative potential inside the neuron can be made either more or less negative depending on the stimulus. If potential is made sufficiently less negative, it reaches a level called threshold, and an action potential is triggered. During the action potential, the neuron suddenly becomes 20 to 50 millivolts positive inside. Action potentials last a few milliseconds before the cell restores its negative resting potential. The cell membrane of a neuron encloses cytoplasm with various ions dissolved in it. The neuron itself is immersed in a salt solution, the extracellular fluid. The ions of the cytoplasm consist mainly of positively charged potassium ions and large negatively charged organic molecules, such as proteins. Outside the cell, the extracellular fluid contains mostly positively charged sodium ions and negatively charged chloride ions. Since charged particles cannot pass through the lipids that make up cell membranes, they must travel through channel proteins that extend through the membrane. In an unstimulated neuron, only potassium ions can cross the membrane, traveling through specific proteins called potassium channels. Although sodium channels are also present, in unstimulated neurons, they remain closed. Since only potassium ions can cross the membrane, and potassium ions are more concentrated inside the cell, they diffuse out of the cell, leaving the large negatively charged organic ions behind. As more and more positively charged potassium ions leave, the inside of the cell becomes increasingly negative. But since opposite charges attract, as potassium ions diffuse out, an electrical force develops that tends to pull them back inside. At some point, the diffusion of potassium ions out of the neuron due to concentration differences is balanced by the electrical attraction tending to pull them back inside. This is the point at which neurons reach resting potential. Reaching resting potential in this way does not require significant changes in the potassium concentration inside or outside the cell. Only about 1/10,000th of the potassium ions initially inside a neuron must leave to create a resting potential of negative 60 millivolts. But it is action potentials, not resting potentials, that carry information through a nervous system.
There we go. Okay. All right. One of the things, it looked like that took a long time. All that takes a long time. But the reality is the sodium and the potassium passing back and forth only takes a millisecond. I mean, it takes a very, very short period of time. It's very, very rapid. Action potential refers to the amount of positive ions that are allowed to flood into the stimulated area of the cell through the sodium channels before the sodium channels close. The action potential for most neurons is positive 40 millivolts. When enough sodium ions flood in to reach a positive 40 millivolts, the sodium channels close. Going beyond equilibrium from the outside environment is called the overshoot phase. So what we're dealing with is it's negative 70 on the inside. As soon as it fires, it gets to the point of positive 40. That's a change of 110 millivolts. And of course, it does overshoot before it can recover itself and everything can go back to normal. They can go back to its resting potential. While a neuron is recovering from stimulation, it cannot be stimulated, and this is known as the refractory phase. This sounds like it takes a long time. All these things, the ions are moving back and forth, but the reality is that it's a very, very short period of time. So the refractory phase is where they are, it is also a very, very relatively short period of time. During the refractory phase, the sodium channels have closed, shutting off the supply of sodium ions flooding into the neuron, and potassium channels open. Because the electrical charge outside the cell is more negative than inside the cell, potassium ions are drawn out of the cell. With the positive, and we just saw this in the video, with the positive potassium ions now flooding out of the cell quickly loses its positive charge, and it not only reaches its resting phase potential, but it goes beyond it in a phase known as the undershoot phase. So there's an overshoot and then there's an undershoot. The potassium leak channels now allow potassium to re-enter the cell to return to the area to its resting potential. Neuron dendrites and somas usually have a different type of membrane than axons. Dendrites and somas usually have sodium channels that are stimulated by chemicals and not by voltage. In other words, they are only activated when the chemical neurotransmitter is present. The axon, on the other hand, does have voltage-gated sodium channels, so the axon responds to stimulation from the dendrite and the soma. Neurons in the central nervous system are myelinated by oligodendrocytes, which are glial cells, of course. And neurons in the peripheral nervous system are myelinated by Schwann cells, which are another type of glial cell. The myelin recovering of the myelin covering of the neuron does not allow potassium leakage from the area covered by the myelin. The depolarization voltage is channeled down the axon from one node of Ranvier to the next, increasing the response by as much as 15 times. This form of conduction is known as saltatory conduction. And as you can see, there's the nodes of, I don't have a pointer. Nodes of Ranvier, as you can see, are the empty spaces where there are no glial cells. Multiple sclerosis is an autoimmune disease where, due to a genetic flaw or environmental trigger, an individual's immune system destroys the myelin on select neurons. This causes a slowing of motor movements and the loss of sensory ability due to the slowing of the neuronal response. Most synapses are triggered by a chemical neurotransmitter in the synaptic cleft, but researchers have discovered that some synapses in the brain are stimulated by electrical responses from another neuron. This is able to occur because the synaptic cleft of these neurons is 1/10 the distance of a chemical synapse. As you can see, the electrical charge can pass from one to the other because they're so close. Not all neurons cause their connection neuron to fire. Some neurons are inhibitory neurons, and instead of raising the excitatory potential, depolarizing the neuron, it lowers the excitatory potential by hyperpolarizing the neuron, making it more difficult for the neuron to fire. And we're going to tell you how that happens right now. Inhibition is done by inducing channels in the receiving neuron to open, allowing more negative chloride ions into the neuron. Since the chloride ions have a negative charge, they lower the neuron's potential to fire. An inhibitory or excitatory neuron is determined by the neurotransmitter that they produce. Some neurons have hundreds of other neurons feeding into them. With these neurons, they may require a number of neuronal stimulations before their threshold is reached. This accumulative effect is required for stimulation is known as spatial stimulation because it has to do with space, the number of neurons firing into that space. Think of this as pain. You smash your finger. You don't hit it all that hard, so it really doesn't hurt. Well, you haven't stimulated the pain. If you hit it hard enough, of course, then you're going to stimulate the pain, and that's all those neurons firing. If you only get 5 or 6 of those neurons firing, it's not going to stimulate it. But if you get 100 of those neurons firing, then of course you know that you've hurt your finger. Some neurons that require an increased threshold will not react from a single neuron stimulation. However, if that single neuron fires in rapid succession, the accumulative effect of the neuronal stimulation will stimulate the neuron to fire. Because it is a matter of adequate stimulation in a select time period, this type of stimulation is called temporal stimulation. And temporal, of course, means time. It's Latin for time. Since a threshold has to be achieved before a neuron will be stimulated, there are various structural factors that can increase the likelihood that a neuron will be stimulated. The number of dendrites receiving stimulation increases the probability that the threshold will be reached. If a glial cell overlaps the synaptic terminal, it may increase the amount of neurotransmitter in the cleft by not allowing leakage. A chemical synapse works in the following manner. The electrical charge travels down the axon to the axon terminal. The charge changes the permeability of the calcium channels, allowing calcium to enter the synaptic terminal. The calcium causes the synaptic vesicles to fuse with the terminal membrane, releasing the chemical neurotransmitter contained in the vesicles into the synaptic cleft. So we've got the calcium. So the chemicals that we're talking about are sodium potassium chloride, which make up, you could have potassium chloride as salt, Sodium chloride is salt, and then you have calcium. The calcium is released by the excitation of the neuron. So calcium, sodium, potassium, and chloride are extremely important chemicals in our bodies. One of the reasons that if you everything tastes like salt on the human body. Practically everything tastes like salt, sweat and whatnot. Mucus, if you've ever, well, of course you've tasted mucus. It's when you blow your nose. Everything tastes like salt, and that's because there's sodium and potassium and chloride mixed together. The calcium, of course, all of these are the electrolytes. So if we were trying to determine if somebody had an electrolyte imbalance, one of the things we would measure their sodium, potassium, chloride, and their CO2. CO2 is carbon dioxide, of course, and we would measure their calcium and their magnesium. And these are the substances. This is what's in Gatorade. This is if you need to replace your electrolytes. What they're talking about is you've been sweating so much that you've lost a lot of salt. So you need to drink something that replaces your electrolytes, and that is what Gatorade is made of. So if you're not really thirsty and you taste Gatorade, then it tastes salty. It's not very salty. It's just a little bit salty, but it's got all the electrolytes in it. It's got calcium, it's got magnesium to some extent, and it's got a lot of sodium and potassium and chloride. After the release of the neurotransmitter into the synaptic cleft, the chemical migrates to the receiving neuron and attaches to transmitter receptors that either inhibit the potential of the receiving neuron or stimulates the neuron to reach its threshold and fire. Much more neurotransmitter is released in the synaptic cleft than can or will be used. So you can't just leave it in there. That's not good. Getting rid of excess neurotransmitter in the synaptic cleft, there's two ways to do it. If you leave it in there, of course, the neuron will spasm. So you need to remove it. The amount of neurotransmitter is detected by the autoreceptor and triggers a stop to the release of the neurotransmitter. Now it's stopped releasing it, but now we need to get it out of there. Removing excess neurotransmitter from the synaptic cleft is done in one of two ways. Either an enzyme in the cleft breaks down the excess neurotransmitter, or receptor sites in the transmitting neuron bind with the neurotransmitter and reuptake the neurotransmitter. Neurotransmitter that is taken back into the neuron through reuptake is recycled by the neuron. And this is how Prozac works. Prozac is a selective serotonin reuptake inhibitor. In other words, it inhibits the reuptake of serotonin, leaving more serotonin in the synaptic cleft. Since serotonin makes you feel not happy, but it makes you not feel unhappy, I guess. Since that's what it does, if we can block its reuptake, we will allow more serotonin to be in the synaptic cleft, and therefore the individual will not be depressed. That's why we do that. Acetylcholine, of course, we don't want too much acetylcholine in there. So one of the things that will happen is the body will send an enzyme into the synaptic cleft to break down the acetylcholine, and the enzyme that it sends in there is acetylcholinesterase. I think I'll say that later on. The neurotransmitter binds to a receptor molecule on the receiving neuron. Each neurotransmitter or ligand has different receptor molecules that they bind to and they are called a ligand binding site. So neurotransmitters, another name for a neurotransmitter is a ligand, and of course they have ligand binding sites. And it's just like this. You can see the red dots there. They are of special structure. What we're really talking about are proteins in the body, and the proteins have receptor sites that only those proteins can bind to. When an excitatory ligand is bound to the receptor molecule, it opens up a sodium channel in the receptor molecule, allowing sodium to enter the neuron. If enough receptor molecules are stimulated by an excitatory ligand, the neuron's threshold will be reached and the neuron will respond with an electrical charge. If the neurotransmitter is an inhibitory ligand, it will open up a chloride channel in the receptor molecule and chloride will enter the neuron. Neurotransmitters are chemicals, and that's one of the things that you need to remember. While each receptor molecule binds exclusively to their neurotransmitter, there are similar chemicals that are structurally similar to the neurotransmitter that can bind to the receptor molecule as well. Acetylcholine is an excitatory neurotransmitter, but the poisons curare from the South American plant and bungarotoxin from the bungaroo snake of Taiwan bind to the receptor molecules, causing paralysis. Now, normally, acetylcholine allows you to move your muscles, allows you to move around, but curare, of course, binds to those receptor sites, and now the acetylcholine can't allow those muscles to move. The bungarotoxin, the poison that the bungaroo snake of Taiwan injects into the individual, it inhabits those receptor sites, and now the individual is paralyzed. Normally, if you're paralyzed, one of the areas that is paralyzed are the muscles of the heart and the lungs, and once those are paralyzed, then your heart stops beating and your lungs stop taking in oxygen, and now that creature is dead. So acetylcholine receptor molecules are also the targets in the case of the poison muscarine from the Amanita muscaria mushroom, and that's what the Amanita muscaria mushroom looks like. It's kind of a cute thing. It's got a red top with white dots on it. Muscarine, however, binds to the receptor molecule and mimics acetylcholine, causing the neuron to fire, resulting in spasms to seizures to death. So we've got bungarotoxin, we've got curare that paralyze you by inhabiting the receptor sites and not allowing the acetylcholine to cause your muscles to move. And we have muscarine. We have muscarine from the Amanita muscaria mushroom that binds to the receptor sites and causes the muscles to keep stimulating, causes the neurons to keep stimulating, and you go into spasms because the neuron just keeps firing. Eventually you have seizures, and then you die because your neurons continue to fire. Chemicals that mimic neurotransmitters. Actually, what happens with these seizures is you can't get your breath because your muscles are spasming so much. The muscles around in your lungs and your heart are spasming, and it causes you to die. Chemicals that mimic neurotransmitters that cause a similar reaction are known as agonists. Chemicals that mimic neurotransmitters and block the action of the neurotransmitter are called antagonists. And of course, the antagonists in this case would be the curare and the bungarotoxin, and the agonist would be the Amanita mushrooms. And here we are going to have, wait a minute, since I don't have that, let's do this again, and we will do this. And now we will see some nurses telling us how this works.
Our presentation is on agonists, antagonists, and receptors. A receptor is a chemical structure on the outside of a cell where an agonist or antagonist binds in order to send the cell a message. An agonist is a molecule that can activate a receptor to send the cell a message. An agonist is non-competitive, binds irreversibly, or competitive, binds reversibly.
Drop it.
Hi, we're talking about receptors. A receptor is like a keyhole in a door, and an agonist will bind like a key and then go into the door, and you will be able to go inside, like a message.
agonist, a molecule that binds to the receptor to block the agonist or antagonist and stop the message from being sent to the cell.
Hi. An antagonist is like the wrong key that will go into the keyhole, but it will not turn. Therefore, the message cannot be sent. I guess we're at the wrong house.
Okay, this information is important for nurses to know because you may be giving drugs that can act as an agonist, which will promote a physiological response to continue, or you might be giving an antagonist, which will make a physiological response stop. An example is when a person has an allergic reaction, they're producing histamine, which is the agonist, and you want to give them an antihistamine, which is the antagonist and stops the allergic reaction.
Ha-choo! Here's some Claritin, which will stop that allergic reaction from happening.
Thank you, nurse.
You're welcome.
I just thought that was kind of cute. Anyway, okay, so I'm sure you learned a lot from the nurses. Okay. Acetylcholine molecule receptors are referred to as cholinergic. Acetylcholine has at least 4 functions and binds to at least 4 cholinergic receptors. Acetylcholine acts on the neuromuscular system as an excitatory neurotransmitter. Acetylcholine acts on the vagus nerve and the heart muscle as inhibitory neurotransmitters, slowing the heart rate down. Acetylcholine is used by the brain in the transfer of memory from short-term to long-term. Acetylcholine acts as an inhibitory neurotransmitter to induce sleep. So acetylcholine is extremely important. It allows us to move, it allows us to remember things, it allows us to go to sleep, and it slows down our hearts when our hearts are beating way too fast. Acetylcholine shares some of its receptor sites with other chemicals, and for this reason, the receptor sites are named after the alternate chemicals. For example, nicotine binds to acetylcholine sites in the skeletal muscle and autonomic ganglia, and for that reason, these sites are referred to as nicotinic cholinergic receptors. Muscarine cholinergic receptors also exist in the autonomic nervous system. Muscarine kills by stopping the heart and paralyzing the intestines. There are 2 types of molecule receptors, ionotropic and metabotropic. Ionotropic, which means ion-moving receptors, open ion channels and allow them to pass through. Ionotropic are very, very fast. Metabotropic, which means change-moving receptors, activate molecules known as G proteins. G proteins bind to the amino acid guanine in its many combinations. These guanine compounds may themselves open the ion channel or be part of a chain of reactions that open the ion channel in turn. And this is really what we were seeing with the first video. They were talking about all these protein activity, all this protein activity. Well, they were talking about mostly they were talking about metabotropic receptors. The metabotropic receptors tend to be relatively slow. Ionotropic is very, very fast. So if we're taking a medication and the medication is ionotropic, it'll get into our systems very, very rapidly. If it's a metabotropic reaction, then it will be relatively slow. And this is important, and it's not important if we're having spasms in our heart and we want to slow it down. One of the things that we can do is take nitroglycerin. The nitroglycerin is ionotropic, and it will stop the spasming almost right away. It will stop the pain, actually, is what it does, because it's ionotropic. However, if we're taking blood pressure medication, blood pressure medications tend to be metabotropic, so they work a lot slower, and that's what we need. We need this reaction to last for an extended length of time, so that our blood pressure won't just go down right now and then go back up. and we need it to be slower for an extended length of time. There are 4 types of neuronal circuits: neural chains, positive feedback circuits, negative feedback circuits, and oscillator circuits. The simplest neuronal circuit is the neural chain. A sensory neuron is activated by a stimulus, and it in turn activates a motor neuron that responds to the stimulus. This is a very simple circuit. It's also fairly rapid. In a positive feedback circuit, the effect is to increase an activity or keep it going. In a negative feedback circuit, the effect is to stop an activity. In an oscillator circuit, the circuit serves as both a positive feedback circuit and a negative feedback circuit, depending on the circumstance. Most sensory neurons collect data, converge the input through a single neuron, and then diverge the signal when it arrives in the brain. The eye does this. Each moment of sight involves over 100 million receptor neurons that converge the information into the optic nerve. It transmits it to the brain so that the signal can be diverged into various parts of the occipital lobe for interpretation. And that's what's going on. If you're looking at your computer right now, you're getting these hundreds of millions of signals on a continual basis, and that is telling you what you're looking at. The electroencephalogram, the EEG, is a device that reads the electrical impulses put off by the neuronal activity in the brain. EEGs are used to map brain functioning and can be used for sleep analysis or to monitor abnormal seizure activity. In case of a coma, the EEG will detect lack of activity or whether there is any activity at all. If we're looking at somebody who is brain dead, what they're really talking about is the fact that their EEG is a straight line. They're not getting any activity at all, or rare activity. You can have a straight line, you can be in a coma, you can be brain dead, and there will still be activity to some extent. Usually they are it's almost like an epileptic seizure. They will just have electrical impulses that shoot through their brain from time to time. It doesn't mean that they're lucid. It means that it just means that the brain is keeping itself alive. 30 million people worldwide suffer from epilepsy. Epilepsy is characterized by seizures. Seizures are involuntary synchronized excitation of large groups of nerve cells in the brain. Seizures can be triggered by trauma or injury to the brain, chemical changes derived from metabolic faults, exposure to toxins, and mutations in ion channels. Epilepsy has to do with sudden electrical charges, and that causes the individual to go into seizures because the neurons are firing and they will not stop firing. Epilepsy is characterized by various types of seizures. There are 2 basic types of seizures. The grand mal seizures are the most extreme form of epilepsy. Grand mal seizures are characterized by total muscle contraction followed by alternate jerking and relaxation. The seizures normally only last 1 to 2 minutes, but recovery may take as long as several hours. Petit mal seizures are characterized by losses of awareness of from 5 to 15 seconds. Complex partial seizures do not involve the entire brain. The individual may experience an aura, a perceived sensation that precedes the seizure. Certain personality traits often accompany complex partial seizures, but it is not certain whether the trait triggers the seizures or the seizures trigger the traits. And this is what it looks like on I think this is a PET scan. Do I have? No, I don't. Okay, just a second. Okay, these things right here, this is where the seizure is occurring. And as you can see, this is when the individual isn't having a seizure, and this is when they are having a seizure. There's a great deal of electrical activity right here in the brain, and it's not going away. And that's causing a seizure for an extended length of time. This is the brain when it isn't having a seizure, and as you can see, there are It's almost like a lightning storm in the brain. And because of that, the individual is having a seizure, and the seizure won't go away until the electrical activities stop. Some individuals are born with these genetic problems, and they will have seizures every 30, 50, 70 seconds, whatever, once an hour, whatever. And these individuals, one of the ways that they can stop this is with CBD oil, which is in marijuana. The CBD oil seems to slow the brain down. As anybody who has been, if you've ever been around somebody smoking pot, that's the way it seems to go. So you could use CBD oil. The other thing that happens, if this is a continual process, since this portion of the brain is malformed, there's something wrong with it. What they'll do is a hemispherectomy, if they are young enough. If they're under the age of 12, what they can do is they can take this portion of the brain out and just leave the other portion of the brain, the other hemisphere of the brain. Now, the weird thing is, if we can get a hold of these kids before they reach the age of 12, they will develop normally, and they'll have minor fine movement problems, but other than that, they're completely normal. Their intelligence, their IQ seems to be about as it would have been. They have normal intelligence. They have normal movements. So we can actually cure epilepsy by taking out half the brain, if it's a constant thing. And of course, that's what the doctors and a lot of times kids will grow out of epileptic seizures. I've had friends that were epileptic. Of course, the problem with epilepsy, you never know when you're going to have a seizure. So most states do not allow these people to drive, because if they have a seizure while they're driving, they could wreck and kill themselves, kill other people, and that's not a good idea. Anyway, so that's what epilepsy looks like. As you can see, it is the fuchsia spot, which is an electrical storm in their brain, as weird as that is. And that is the end of the chapter. And so I'm going to go